Structure and function of the cardiovascular and lymphatic systems. The circulatory system is divided into two major circuits, the pulmonary and the systemic. The right side of the heart is responsible for pulmonary circulation, sending deoxygenated blood to the lungs. Meanwhile, the left side of the heart manages systemic circulation, pumping oxygenated blood to the rest of the body. These two systems work together to ensure efficient oxygen delivery and waste removal. This diagram provides a visual overview of blood flow through the body. Blood from the systemic veins enters the right atrium, flows to the lungs via the right ventricle, and returns oxygen-rich to the left atrium. The left ventricle then pumps it through the aorta to all tissues. This cycle maintains a steady flow of nutrients and oxygen to every cell. Here we see a continuation of systemic and pulmonary circulation. Notice how arteries carry blood away from the heart while veins return it. The heart acts as a dual pump, carefully coordinating contractions to maintain this directional flow. This foundational layout supports every organ system. The heart is enclosed in the pericardium and composed of three layers: the epicardium, myocardium, and endocardium. The epicardium is the outer protective layer. The myocardium is the muscular middle layer responsible for contractions, and the endocardium lines the interior chambers. These layers work in unison to support strong, rhythmic heartbeats. Surrounding the heart are structures known as the great vessels. These include the superior and inferior vena cava, pulmonary arteries, pulmonary veins, and the aorta. They serve as critical conduits, directing blood in and out of the heart chambers. Each vessel has a specific function in systemic and pulmonary flow. The great vessels are major blood vessels connected directly to the heart. The superior and inferior vena cava bring deoxygenated blood from the systemic circulation into the right atrium. The right and left pulmonary arteries carry this blood to the lungs for oxygenation. Oxygenated blood returns to the heart via the pulmonary veins, and the aorta then distributes it to the rest of the body. These vessels are essential conduits for maintaining circulation through the pulmonary and systemic circuits. The heart contains four chambers, two atria and two ventricles. The atria are the smaller, upper chambers that receive blood, while the ventricles are the larger, lower chambers that pump blood out. The left ventricle has the thickest wall because it must generate high pressure to deliver blood throughout the body. This structure ensures directional and forceful flow. This slide expands on the structural features of each heart chamber. Blood flows from the atria into the ventricles through valves that prevent backflow. The right heart handles deoxygenated blood, while the left manages oxygenated blood. This division is vital for separating pulmonary and systemic functions. The heart's valves ensure unidirectional blood flow. Atrioventricular valves, including the tricuspid and mitral valves, are located between the atria and ventricles. Semilunar valves, the pulmonary and aortic valves, are found between the ventricles and major arteries. These structures open and close in response to pressure changes during the cardiac cycle. Each valve is uniquely shaped to support its function. Atrioventricular valves have flaps that close during ventricular contraction, preventing regurgitation. Semilunar valves feature cup-shaped cusps that seal tightly during diastole. Proper valve function is essential for efficient cardiac output. Blood flow in the heart is controlled by pressure gradients and valve actions. During diastole, the ventricles relax and fill with blood. In systole, the ventricles contract, increasing pressure and forcing blood out. The cardiac cycle ensures that each heartbeat delivers a measured amount of blood to the lungs and body. This diagram reinforces the timing and directionality of blood flow during the cardiac cycle. As pressure builds in the ventricles during systole, semilunar valves open, allowing blood to exit toward the lungs and body. During diastole, atrioventricular valves open so the ventricles can fill. Proper valve function and pressure regulation ensure efficient heart performance. The phases of the cardiac cycle include atrial contraction, ventricular contraction, and relaxation. Each phase coordinates with specific valve movements and pressure shifts. Diastole allows blood to refill the heart, while systole propels blood outward. A full cycle completes with every heartbeat, repeating about 60 to 100 times per minute in a healthy adult. Coronary circulation supplies oxygen and nutrients directly to the heart muscle. The right coronary artery branches into the conus, right marginal, and posterior descending arteries. The left coronary artery divides into the left anterior descending and circumflex arteries. These vessels ensure the myocardium stays well oxygenated, especially during increased demand. Coronary capillaries are small vessels that deliver nutrients directly to heart muscle cells. Blood then drains into coronary veins, including the coronary sinus, great cardiac vein, and posterior vein of the left ventricle. Lymphatic vessels also assist by removing excess fluid. These combined pathways help maintain a balanced environment for cardiac tissue. This final view of coronary circulation highlights its critical role in cardiac performance. Obstruction in any coronary artery can lead to ischemia or myocardial infarction. Understanding this system is essential in diagnosing and treating coronary artery disease. The heart's reliance on continuous blood flow cannot be overstated. The heart's electrical system regulates rhythm and timing. The sinoatrial or SA node initiates electrical impulses, followed by the atrioventricular or AV node. Signals travel through the bundle of His and Purkinje fibers, triggering coordinated contractions. This conduction system maintains a steady, synchronized heartbeat. This slide illustrates the pathway of electrical impulses across the heart. Each node and fiber plays a role in depolarization and contraction timing. The coordination ensures the atria contract just before the ventricles, allowing complete ventricular filling. Any disruption can lead to arrhythmias and reduced cardiac output. Cardiac action potentials initiate and regulate the heart's contraction cycles. These include depolarization, repolarization, and refractory phases. Automaticity allows pacemaker cells to fire without external stimulation, while rhythmicity ensures a regular pace. These electrical properties are crucial for proper heart function and interpreting ECGs. The electrocardiogram, or ECG, is a non-invasive tool used to measure the electrical activity of the heart. It captures the sequence of depolarization and repolarization across the heart muscle. Different waveforms on the ECG correspond to specific events in the cardiac cycle, like atrial and ventricular contraction. ECGs help detect arrhythmias, ischemia, and other cardiac abnormalities. The autonomic nervous system plays a major role in controlling heart rate and contractility. Sympathetic nerves increase the speed and force of contractions, enhancing cardiac output. In contrast, parasympathetic nerves, primarily via the vagus nerve, slow the heart rate and reduce contractility. Together, these systems balance the heart's performance based on the body's needs. Myocardial cells resemble skeletal muscle cells, but have distinct features essential for heart function. They contain sarcomeres and contractile proteins like actin and myosin, but also possess intercalated discs that facilitate electrical connectivity. These cells rely heavily on oxygen and have abundant mitochondria to meet their constant energy demands. Their unique structure enables coordinated, sustained contractions. Cardiac contraction begins at the level of the sarcomere the muscle's functional unit. When myosin bridges attach to actin filaments, the sarcomere shortens, causing muscle contraction. This process is part of excitation-contraction coupling. The heart's ability to pump depends on the efficiency of these molecular interactions. During relaxation, calcium ions are actively pumped back into the sarcoplasmic reticulum. This allows the myofilaments to return to their resting state. Proper calcium handling is critical for preventing fatigue and maintaining rhythmic contractions. Any disruption in this process can lead to cardiac dysfunction. The heart's unique muscle architecture supports continuous contraction and relaxation without fatigue. This slide emphasizes the role of ATP and calcium in cycling contraction and relaxation. Myocardial efficiency depends on a steady energy supply and precise ion regulation. These processes are tightly regulated to sustain lifelong function. Cardiac output is the volume of blood the heart pumps per minute, calculated by multiplying heart rate and stroke volume. In a healthy adult, this is about 5 liters per minute. Ejection fraction, the percentage of blood ejected from the ventricle per beat, is another key measure of heart performance. Efficient cardiac output ensures adequate tissue perfusion. Several factors influence cardiac output. Preload is the volume in the ventricle at the end of diastole, while afterload is the resistance the heart must overcome to eject blood. The Frank-Starling law states that the heart pumps more forcefully with greater stretch. Together, preload, afterload, and contractility determine how effectively the heart meets the body's needs. Myocardial contractility is influenced by preload, stroke volume, and inotropic agents. Factors like oxygen and carbon dioxide levels can modify how forcefully the heart contracts. Heart rate also plays a role. Too fast or too slow, and cardiac output may decline. Neural reflexes and hormones fine-tune these variables for balance. Systemic circulation delivers oxygenated blood from the left ventricle to the body and returns deoxygenated blood to the right atrium. It involves arteries, arterioles, capillaries, venules, and veins. Each vessel type has a unique structure that supports its role in circulation. This system is essential for tissue oxygenation and nutrient delivery. Blood vessels are composed of 3 main layers: the tunica intima, tunica media, and tunica externa. The lumen is the central cavity through which blood flows. Endothelial cells line the vessel walls, playing key roles in coagulation, immune response, and vasomotion. These structures support both stability and dynamic responsiveness. The tunica media is primarily made of smooth muscle and elastic tissue, allowing vessels to constrict or dilate. The outermost layer, the tunica externa, provides structural integrity. Small blood vessels called vasa viscorum supply nutrients to large vessel walls. This architecture enables vessels to endure pressure changes. Arterial vessels carry oxygen-rich blood away from the heart. They range from large elastic arteries to smaller muscular arteries and arterioles. Capillaries are the smallest vessels where gas and nutrient exchange occurs. This branching network ensures delivery to every cell in the body. Venous vessels return deoxygenated blood to the heart. They include venules, veins, and large central veins like the vena cava. Unlike arteries, veins have thinner walls and valves to prevent backflow. Their structure accommodates large volumes of blood at low pressure. Blood flow is driven by pressure and opposed by resistance. Resistance depends on vessel diameter and length. Narrower vessels increase resistance. Poiseuille's law describes this relationship mathematically. These factors collectively determine how easily blood moves through the circulatory system. Velocity refers to the speed of blood flow and is affected by vessel size. Vascular compliance describes a vessel's ability to stretch and accommodate blood. Laminar flow is smooth and efficient, while turbulent flow is chaotic and increases resistance. Proper flow patterns are vital for efficient circulation. Laminar flow occurs in parallel layers with minimal friction, while turbulent flow is disorganized and noisy. Turbulent flow can be caused by vessel narrowing, high velocity, or obstructions. It's often detected in clinical exams as a murmur or bruit. Understanding these patterns helps in diagnosing vascular abnormalities. Blood pressure is primarily controlled through arterial pressure and venous return. Systolic pressure reflects the force during ventricular contraction, while diastolic pressure reflects relaxation. Mean arterial pressure, or MAP, provides an average level of perfusion across the cardiac cycle. Venous pressure is influenced by fluid volume and vessel wall compliance. Blood pressure is affected by cardiac output and total peripheral resistance. Baroreceptors detect changes in pressure and send signals to the brain to adjust heart rate and vessel tone. Chemoreceptors respond to chemical changes like oxygen or carbon dioxide levels. Hormones and mediators like nitric oxide and endothelium also influence vascular tone. Baroreceptors are stretch-sensitive receptors located in the aorta and carotid arteries. When blood pressure rises, these receptors stimulate the parasympathetic nervous system to slow heart rate. When pressure drops, they activate the sympathetic system to raise heart rate and constrict vessels. This reflex helps maintain homeostasis in real time. Chemoreflexes respond to low oxygen, high carbon dioxide, or low pH. These signals trigger increased heart rate and vasoconstriction to improve oxygen delivery. Located in the carotid and aortic bodies, these receptors work closely with respiratory centers. They are essential during conditions like hypoxia or acidosis. Coronary circulation is regulated by perfusion pressure and autoregulation mechanisms. The heart increases blood flow during high demand, such as exercise or stress. Autonomic regulation helps balance supply with myocardial oxygen needs. Efficient coronary circulation is essential to prevent ischemia or infarction. The lymphatic system is a network that returns excess fluid from tissues back into the bloodstream. Lymph, a clear fluid, flows through lymphatic veins, ducts, and nodes. The right lymphatic duct and thoracic duct are main drainage points. This system supports immune defense and fluid balance. Lymph nodes filter pathogens and house immune cells. Afferent vessels bring lymph into the node, and efferent vessels carry it out. These checkpoints are critical in detecting infections and triggering immune responses. The lymphatic system works closely with blood vessels to maintain internal homeostasis.